Hello and welcome to Genetics Day 5 Classwork. So we're actually going to be doing this during class on Zoom, but it is a little long and it's a little complicated. So I'm also going to be making this video of me going through the assignment just in case you missed class or you're confused and you want to go back and see what the heck happened. So how does DNA code for protein synthesis? So we're going through all of the steps of protein synthesis here. So we're going to go from transcription to translation to the actual translation part, which is taking the messenger RNA codons and figuring out which amino acids they code for, which is going to involve us using this universal genetic code chart. So let's go ahead and get started. And again, we are going to be doing this during class on Zoom. But if you don't finish it during class or you're confused about what we do during class, you can watch this video to help you understand what just happened. So instructions. You will help complete the major steps of protein synthesis. So we're going to be like the enzymes assisting the process. So transcription, going from DNA to mRNA, translation, where mRNA is translated into amino acids using tRNA, and then the actual protein synthesis, where the amino acids are put in order to build a protein. So, transcription. DNA carries the genetic instructions or code to produce proteins. The code is actually the sequence of bases in DNA. For example, ATAGCGA is a code for part of a protein. The entire protein would actually require a much longer sequence of DNA bases. Whenever a protein needs to be made, the correct DNA sequence for that protein is copied to a molecule called messenger RNA. So, let's go over that last part again for emphasis. Whenever a protein needs to be made, the correct DNA sequence for that protein is copied to a molecule called messenger RNA. Since DNA is so important, it is efficient to simply copy its code when needed and leave the original safe in the nucleus, right? Because DNA is a very large molecule, and most of the time when it needs to be copied, it's only part of a DNA molecule, perhaps that codes for one gene that needs to be used to synthesize the appropriate protein. So lots of copies can be made at any time. Lots of copies can be made of DNA to produce new cells. Lots of copies can be made into RNA to produce many proteins. The process of copying a sequence of bases in DNA into a complementary mRNA strand is called transcription. It occurs in the nucleus and is the first step of protein synthesis. So let's go ahead and get started. Remember, in this case, instead of A pairing with T, it pairs with U, but G still pairs with C. And that also means that T is still going to pair with A, right? Because we're starting with the base DNA. So there's still going to be thymine in that DNA. So let's go ahead and do some pairing. G pairs with C, A pairs with U, A pairs with U, C pairs with G. We can divide these into codons. 3 codons. And here, for those of you that might be confused, I'm actually going to color code this for you. So all of our C's and G's are going to be green. And all of our A's and U's are going to be red. T still pairs with A, right? Because adenine is still present in mRNA. But A pairs with U, and again, T pairs with A. So we have 3 codons here, CUU, GAG, UCA. Which base in RNA is replaced by uracil? Well, we just went through this, but that's thymine, baby. How many mRNA codons are illustrated above? 3, right? And we can tell because codons are triplets, groups of 3, 1, 2, 3. Count them. 1, 2, 3. What is the name of the enzyme that creates the mRNA copy from the DNA? Just like when DNA is being replicated, DNA polymerase comes in and brings in the appropriate nucleotide bases. When mRNA is being copied from DNA, that's RNA polymerase. And we can tell it's an enzyme because it ends in ASE. What is the name of the sugar in the mRNA nucleotide? So in DNA, it's deoxyribose, but in RNA, there's no D, it's ribose. Let's put that in a different color. Ribose. And that is the sugar in the sugar phosphate background of the double helix structure, which is this part, the outside part that makes up the backbone of the DNA molecule. And then we have the alternating, or maybe sometimes repeating several times, bases on the inside. So that is the sugar phosphate backbone of the DNA or RNA strand. And this double helix represents DNA, right? So DNA is double stranded, while RNA is just one single strand. And that is important. So the DNA is unzipped to just be a single strand. One half of the double helix. And then that single-stranded DNA is used as a template to produce single-stranded RNA. So RNA is created using one half of the DNA molecule, like so, to produce a single strand of RNA, which can then safely leave the nucleus and be used to produce proteins in translation. So what is the mRNA transcript for the DNA sequence TTA-CGC? So we're going to pair them just like we did in the first step up here. Right underneath, T pairs with A, T pairs with A pairs with U, C pairs with G, G pairs with C, C pairs with G. So we have A, U, G, C, G, and that is 2 codons. So now translation. Now that an mRNA copy has been made, the DNA zips back up and can be used again later to make more copies. So then it goes back into its spot in the nucleolus and is chilling. Now the mRNA can leave the nucleus through pores in the nuclear membrane and head to the protein factory of the ribosomes. Remember that the mRNA copy is also a genetic code or how to make a protein. The code was originally copied from the DNA, which is much too valuable to be used itself to make a protein, right? The DNA can't just leave the nucleus and go to the ribosome, but the mRNA can. So a copy was made called mRNA, which stands for messenger RNA. The mRNA moves to the cytoplasm where the code will be translated on a ribosome by transfer RNA, tRNA molecules. Each tRNA, and tRNA, just like mRNA, has uracil as a base instead of thymine. Each tRNA carries a specific amino acid, which will be needed to build the protein. Since a protein consists of a precise sequence of amino acids, the right tRNA must insert its amino acid into the chain when needed. But how does this happen? So let's just emphasize the most important part of this here. Each tRNA carries a specific amino acid which will be needed to build the protein. So you can think of it, the mRNA is the messenger that is carrying the message how to make the protein out of the nucleus to the ribosome, and then the transfer RNA is transferring the correct amino acids in order to build the protein structure. On one end of the tRNA molecule is the amino acid, and on the other end is a set of 3 bases called an anticodon. So we depict the tRNA like this, right? A little T, like a little plus sign. And then on the end is the amino acid. The 3 anticodon bases, the 3 anticodon bases use complementary base pairing with the 3 mRNA bases called a codon. So there's the codon, and then there's the anticodon, which is its complement. And if they fit, this is the correct tRNA molecule and therefore the correct amino acid. See how the tRNA anticodon CGA is complementary to the mRNA codon GCU. Note that both tRNA and mRNA have uracil instead of thymine. Since the tRNA molecules are essentially reading the mRNA code by binding to it and bringing in the proper amino acids to build proteins, this process is called translation. So the tRNA is translating from the language of RNA into the language of amino acids, right? So the language of genetic code So the first part is transcribing one language of genetic code DNA to another language of genetic code mRNA. And the significant thing about that mRNA, it's very similar to DNA, but it can leave the nucleus, right? So that is transcription. Or like when you hear something and you write it down, you're transcribing it, that is transcription. But then translation is sort of changing languages, taking from genetic code language to protein language, and amino acids are the building blocks of these proteins. So I'm already sensing this video is getting a little long. So what I think I will do is I will go up until this question right here, and then I'll film you a part 2. So let's do this bit here. So again, we're going now from RNA to RNA language, so there's going to be no thymine involved. C pairs with G, U pairs with A, U pairs with A, G pairs with C, A pairs with U, G pairs with C, U pairs with A, C pairs with G, U pairs, or A pairs with U. And again, we're going to split these up into codons because we're going to take these codons and using the universal codon chart, figure out which amino acids are coded for in each one. So what organelle assists tRNA in translating the mRNA in the cytoplasm? Why, it's the ribosome, of course. The role of tRNA is to carry an amino acid. Is a tRNA anticodon more similar to DNA or RNA in nucleotide sequence? RNA. Also because it uses U instead of T, right? There's no thymine. If the mRNA codon is CGA, then the tRNA codon that binds with it, or the tRNA anticodon that binds with it, so again, we're just going to follow our base pairing rules again. C pairs with G, G pairs with C, A pairs with U. GCU. Shwaboom. All right, so then this next step, protein synthesis, and how to actually use this universal codon chart, I'm gonna create a Unit 5 Genetics Day 5 classwork Part 2 to actually finish translation. I hope you enjoyed that and that made sense to you. Stay tuned for Part 2.